to accumulation of pharmaceuticals in the environment and on human health. I'm sorry for inconvenience. Yeah. This is this slide I'm showing the threats to human exposure. As we know that long-term exposure to these contaminants that may result in acute and chronic damages. And the most important, the recent discovery is the change in the behavior of the offsprings of those mothers who are over-consuming the extra doses of paracetamol. That is basically bringing up the behavioral disorders in the offsprings. That is, I've already discussed that ADHD and OSD. And I just wanted to point out that my son is also suffering from the same diseases. So next come the ecotoxicity of paracetamol. Ecotoxicology, these studies are basically studying the effect of the toxic nature of these pharmaceuticals on aquatic life forms, on the sediments and the soil life forms. It is found in many research that these toxicants actually alter the growth, reproduction, and the survival abilities of these forms. For these ecotoxicological studies, that is done with the help of model organisms, and the model organism should be selected on the basis of genetic, physiological, morphological, molecular, and ecological traits. The model organism should be, the chosen model organism should be inexpensive in nature. It should be comfortable to be maintained in laboratory conditions, and it should be fast growing and easily reproducible, and it should be highly characterized in nature. Like for example, zebra fish is the most commonly and the most often used model organism for these ecotoxicological studies. And these studies are essential because they provide information related to the action of these pharmaceuticals, toxicants, on other related species. And on the basis of the mechanism of action on extrapolated estimation can be made about the surrounding ecology. Those studies help us to identify the root cause relationship between toxicant and its toxic effects on the overall marine population. These are the environmental risks which we can understand in terms of the most the threatening nature of these pharmaceutical compounds, because they are highly recalcitrant in nature, they are bioactive compounds, and they have the capability to disrupt the endocrine system of especially marine life and fish population. This is an overview of 6 extensively used drugs category, which I have taken from different categories of drugs. The most important, most commonly used drugs have been taken, like from antibiotics, non-steroidal anti-inflammatory drugs, antihypertensives, anti-epileptics, and lipid regulators. I'll be discussing about the overview of the chemical structure, the metabolic rate, and the removal via sewage treatment plants. For example, the first example which I've taken is an antibiotic, which is ofloxacin. Of course, this is these medicines which I've chosen here, 6 medicines, they are extensively used. So I can see from this that excretion rate is not 100%. That remains 70% unchanged, and the same goes with diclofenac, which is in the category of a non-steroidal anti-inflammatory drug. Again, the excretion rate is not 100%, that is 15% remain unchanged. And the data show that the total removal via sewage treatment plants is only 69%, and for ofloxacin, the data is not available. The next is anti-epileptic carbamazepine, and the antihypertensive is propranolol. We can see the same data here. Excretion rate is not 100%. I mean, that is not completely degraded in the human body. So they are somehow going in the form of metabolites, metabolic byproducts into the system, that is environment system. So the total removal is, again, that is only here, this is in case of carbamazepine, this is only 7%. And for propranolol, this is 96%. The next is sulfametazole and clofebric acid. They're antibiotic and lipid regulators. The same data shows that these drugs are not completely metabolized in human body. And at the same time, if we are trying to remove them through sewage treatment plants from the environment, they are not completely eradicated. So now here comes the responsibility, how these pharmaceuticals are detected in the system, the environmental system. So this can be done with the help of the following processes. Like firstly, we can do the extraction of analytes. The next comes out to be detection of analytes, and the last is identification. So the extraction of analytes can be done with the help of Soxtet extraction, superficial fluid extraction, ultrasonic and microwave assisted model methods are used. Basically, all these techniques are used in combination with chromatography and mass spectrometry. And the basic idea is to isolate, to detect, to isolate, to ionize, to detect, to analyze, identify, and report the analyte present. Though it is a fact that detection of pharmaceuticals and the metabolic transformation products need to be modified according to the chemical nature of analyte. So the next comes out to be detection, MS3 technique, which is known as multiple mass spectrometry, which is a very useful technique for the identification of close resembling compounds and the transformation products and isomers. For example, according to a study, MS3 technique, this was used to distinguish a mixture of isochlorotetracycline and a photo-oxidation transformation product of isochlorotetracycline, which were different from each other only by the addition of only one oxygen molecule. These are the current approaches of treatment. Once they are we know that they are being introduced, they are being accumulated, these pollutants are being accumulated in the system. So we are following some certain treatments to remove them. So biological treatment approach, the activated sludge approach, which is the most commonly used wastewater treatment technology. And it is reported that biological treatments, when they are combined with the biosorption and biodegradation influences removal of these organic micropollutants to a greater extent. None of the strategy which is currently available is completely successful to eradicate these micropollutants from the system. But typically, we can achieve a greater level of removal by advanced oxidation processes, photocatalysis, membrane filtration, and adsorption techniques. This is the chart which is showing the different types of advanced oxidation processes, homogeneous and heterogeneous processes. So we can see that heterogeneous processes are done with the help of catalytic ozonation, photocatalytic ozonation, heterogeneous photocatalysis, and homogeneous processes are done with the help of either we can use we can do with the help of energy or without energy, and different methods are shown in the diagram. Now comes to biological treatment. As the name suggests, the biological treatment systems, they have a variety of active microorganisms that can be used to stimulate a wide range of chemical transformations. Many studies have reported that the removal of organic micropollutants occur to a varying extent in biological treatment systems when we combine these systems, biosorption and biodegradation. Therefore, it is very important to implement these techniques on a pilot scale so that we can check the efficiency of wastewater treatment containing pharmaceutical pollutants, whether they are efficient or not. This is the diagram which shows the biological treatment of pharmaceuticals. As we can see, the right side shows that plant bacteria and with a functional gene, that is microorganisms, when they're combined with the bioremediation conventional methods, and the left side shows that they are bringing better results. That brings the increased rate of bioremediation. These are the bioremediation techniques which we are using, different types of natural, intrinsic. So this is, I don't think I would be able to explain such a short time. These are the challenges and the limitations with the current treatment technologies of removal of pharmaceuticals. Many, as I discussed, that advanced oxidation techniques, they are to a greater extent, they are able to remove these contaminants from the environment. Like advanced oxidation processes include ozonation, activated granular carbon, UV radiation, membrane technology. They are used with relative success, but there is a category of different pharmaceuticals, like I discussed just now: antibiotics, anti-epileptics, non-steroidal anti-inflammatory drugs, regulators. They are known to resist these treatments because they're highly soluble in nature, and they are poor degradable in water. So these are the techniques. How can we enhance the biological wastewater treatment? So this can be done by exploiting microbial diversity, exploiting metabolic diversity, exploiting biocatalyst diversity. So exploiting microbial diversity, this is basically done with the help of oxidative enzymes. They have great potential as biocatalysts for breaking down of organic waste. One of the most common, the most prominent example is oxygenase cytochrome P450, which is a highly efficient group of monooxygenases, which is responsible for the destruction of drugs and toxins in microorganisms. And then the second class is lacases, which is a group of copper-containing oxidative enzymes, which is used for the microorganisms to break down the lignin. Both these enzymes have been shown to efficiently degrade a wide variety of organic micropollutants. So the next point comes out to be exploiting metabolic diversity. Like many enzymes which are promising biocatalysts, they are capable of transforming a variety of substrates. These enzymes are basically generalized in nature and they're not essential. So they maintain low metabolic flux and they require less regulation than specialized enzymes. The next is exploiting biocatalyst diversity. As these non-specialized enzymes are promising biocatalysts for degradation of organic pollutants. and the non-specific nature make them promising biocatalysts for degradation of organic micropollutant. These are the opportunities which should be used to reduce the input of these pharmaceuticals. I believe that we always say that prevention is better than cure. So why not we should start thinking at our level only, that why shouldn't we reduce the release of pharmaceuticals into the environment with the rational uses? So we can divide the responsibilities in different heads. For example, the first responsibilities comes out to be a pharmacist. They must inform the consumers to how can they, so that they can participate in take back system if appropriate and if it is necessary. The second responsibility is from pharmaceutical companies. They basically they must follow green and sustainable practices. They must publish relevant information for environmental assessment. They must offer suitable pack sizes. They must establish take back system as like pharmacist whenever it is required. So the next responsibilities from science and humanities, these branches must begin research to ban in compounds with green and sustainable pharmacy. They must set regulation criteria for the risk assessment and management of waste. And the last responsibilities that comes out to be of government authorities and agencies which are handling these production software production techniques. So they must initiate communication related to threshold for active pharmaceutical ingredients in different environmental areas, and especially in drinking water. They must establish a restrictive connection so that they must improve their legislation for the management of outdated medicines and for legal and fair uses. And last responsibilities, of course, it is no one but it's us responsibility. So I would like to summarize my presentation here. That finally we have discussed that definitely we are using these pharmaceuticals for our health, for healthcare, for well-being. But this is again a very bare fact that these are present in the form of mixtures, in the form of metabolic byproducts, and in the form of micropollutants in the environment on a global scale. And they are transformed to other toxic end products once they are metabolized in the human body, and they are released into the environment by metabolic pathways. So only the knowledge of their fate and occurrence because of the long-term exposure is actually not sufficient for the assessment of final risk. We must go one step ahead. And there is a knowledge of, there's a lack of knowledge concerning like what is the quantity that is being released and the metabolites which enter the environment. So once we know that, yes, they are persisting in the environment, we must follow certain measures and we must become a part of risk management system and the waste management so that we can minimize the waste from the environment. So these removal techniques we are definitely using, but they are not able to eradicate their occurrence from the global system. And the bioremediation techniques, though they are like done with the help of microorganisms, bacteria, fungal, and the plant species, they are known as the promising bioremediation techniques, and they have great potential with regard to removal of pharmaceuticals. But none of the strategy is completely successful. So we must follow certain latest, we must switch on to some those pathways where we can find the exact solution. How can we, firstly, we can reduce the input, and next the degradation pathways, which are more efficient. So I would like to conclude my presentation here. That since they are present and these technologies are not completely effective, which we are using to remove them. But the one thing is that when we combine physiochemical treatment along with the biological treatment, the results comes out to be better. The next stage for the treatment of wastewater system, that could be the novel metabolic traits for unconventional microbe synthesis of enzymes, which are capable of degrading micropollutants, and a move towards green and sustainable pharmacy. Along with the scientific treatment methods, we must not ignore, I would request you all that we must not ignore the cultural aspects to understand the minimum and the rational uses of drugs so that we can save our environment for the, from the hazardous post effects. These are the references which I've included. Thank you so much.